Right. So in our last class, we discussed about reagents, more particularly reagents used for the preparation or method or methods used for the preparation of peptide or polymers. And also we discussed the differences between the conventional methods and the solid phase peptide synthesis. So solid phase peptide synthesis means a resin will be there. So to that resin, one of the amino acid is bound. More particularly, C-terminal end of the amino acid is bound to the resin. So here, what is the principle is involved? So look here, I'm taking. So this is the resin, and this will be the C-terminal end of the amino acid. So when this C-terminal end of the amino acid is bound to the resin, and you know that resin is not soluble in the solvent, any reaction media, but the part, okay, amino acid part which is attached to the resin, it is soluble in the reaction media. Let's give an entry. Okay. Right. So what I am saying, it's a resin. So when resin is treated with C-terminal end of the amino acid, so there will be a bond formation, a removal of H and Cl, and resin is insoluble in the reaction media, and this amino acid is soluble. So when this amino acid is bound, okay, form a bond with the resin, then a material will be like this. Once there is a bond formation with the resin, this resin is not soluble in the reaction media, but this part, amino acid part, is soluble in the reaction media. In the reaction media, other reactant means whatever the amino acid you want to treat with this, okay, the peptide, which peptide you want to make, and what is the sequence is involved. For example, if this is a glycine, then you can treat with the alanine. So then what you will get? You will get alanyl glycine as the dipeptide. So that means appropriate amino acid, it is taken in the reaction media in excess quantity. So what I mean to say that appropriate amino acid, it will be present in the reaction media. A solid bound amino acid, it is soluble, amino acid part only soluble in the reaction media, not the solid support. When this amino acid part of the solid resin react with the amino acid present in the reaction media, once the reaction is completed and once the reaction occurs, so that means deprotection of this Bach group and treated with the free C-terminal, free carboxylic group of the amino acid. So there will be a reaction between the amino group of the what is called as solid supported one react with the free carboxylic group which is present in a reaction media. Once there is a bond formation, that amino acid also is not obtained peptide also is not soluble in the reaction media. So that means it will convert as a solid. So once it will convert as a solid, it was filtered and wash. Wash means the solvent used for the reaction. That wash is given to the peptide. So thereby one can reduce the steps, one can reduce the time consuming processes, repetitive processes. So that is the reason solid phase peptide synthesis as an advantage. Using a solid phase peptide synthesis, one can do deprotection and coupling reaction can be achieved on the surface of the solid support. Okay, so by this way, so this process, what is the process? Deprotection and then coupling. Okay, so when you are coupling, the N is protected. Again, deprotection and then coupling. So like that, we can proceed until getting a desired peptide. The desired peptide may be a tripeptide or pentapeptide or polypeptide. Okay, once the desired peptide is obtained, then what we need to do? So need to release of the peptide from the resin. Okay, then a peptide is obtained. So this is the solid, this is the principle, or this is the process of solid phase peptide synthesis. I think now you are familiar with the what is solid phase peptide synthesis, what is the conventional method of peptide synthesis. Conventional method of peptide synthesis involves blocking, activation, coupling, and then deprotection for every amino acid. In addition to this, there is also one more process called crystallization, one more process called acidification and basification. Okay, all these steps are involved. So conventional method is a repetitive method. Whereas the solid phase peptide synthesis, what is the principle involved in a solid phase peptide synthesis? A amino acid is bound to a solid support. Okay, and this was made to react with a solution. One of the reactant is taken in excess. Once the reaction occurs, so it was washed with the solvent, plenty of solvent, to get rid of the unwanted products. So this is the principle involved in a peptide synthesis. And main thing you need to mention that solid phase peptide synthesis is the one. Here, deprotection and coupling is achieved on the surface of the solid support. So that word is enough, and remaining thing you write it. Okay, so this is the method or principle would involved in a solid phase peptide synthesis. That definitely there are questions on solid phase peptide synthesis. More particularly, so what is the principle involved in a solid phase peptide synthesis? What is Merrifield resin? How it is useful in a peptide bond formation? Okay. So these are the questions one can frame on this particular solid phase peptide synthesis. So now I am taking two examples. So in that two example, I am employing a solid phase peptide method to synthesize a pentapeptide. Okay, so pentapeptide, more particularly, these are called enkephalins. Okay, I will go to the next slide. So next slide will give you the method of synthesis of a pentapeptide, where there is a involvement of solid phase synthesis. Okay, I will go to that. So this is the one. So what is this? This is a methionine. So I am talking about a pentapeptide. So what is the pentapeptide? Tyrosine, glycine, phenylalanine, methionine. So this is the pentapeptide. So if you want to synthesize this pentapeptide in a solution phase synthesis, first you have to take a tyrosine. So tyrosine is having a phenol side chain. The phenol OH group should be protected with benzyl group, and then N protection. Then afterwards, you have to activate the N protected tyrosine and then couple with the glycine. After that, activation of dipeptide and then couple with the glycine. Activate the carboxylic acid and then couple with the phenylalanine. Activate the carboxylic and then treat with methionine, and then deprotection. So this much process is involved in a solution phase peptide synthesis. So that means in a solution phase synthesis, so you are starting with the N-terminal end of the amino acid. So whereas in the solid phase peptide synthesis, you should start with the C-terminal end of the amino acid. So here I am talking about the solid phase peptide synthesis. So I have to take this methionine. How I have to take? Amino group should be protected. So here F mark I am taking. F mark is the N protection used for this method of synthesis, method of solid phase peptide synthesis. So F mark methionine. So it was treated with a resin. This is a solid supported resin. So what will happen? So there should be removal of OH and H. So where H is from the carboxylic acid of the methionine. So methionine carboxylic group is react with this alcohol, acid and alcohol, ester formation. So what is removal? H2O is removal. So you know that DCC is a reagent. It will take the H2O and convert into what is called as DCU. So this conversion DCC to DCU is catalyzed by HOBT. HOBT is a reagent. It's a coupling reagent. So using an HOBT DCC method, it is possible to do a activation as well as coupling on the in a single method. Once there is a removal of H and OH, so ester is formed. So that's what I have written here. So now what is happened? A amino acid is bound to a solid support. So this one is treated with a reaction mass containing phenylalanine. So reaction mass contain N-protected phenylalanine. So before what we need to do? Deprotect this F mark. How the deprotection of F mark? So with 20% pyridine. So 20% pyridine deprotection of F mark. So what is obtained? This should be a free amino group. So I will correct it here itself. 1 minute. Okay, so what you need to do? So this F mark is not there. So this is a free NH2. So you need to make this as a NH2, free NH2. So because F mark is removed here. So 20% piperidine or pyridine means F mark is removed and this is NH2, free NH2. So that means deprotection is one method. And then coupling with the appropriate amino acid. So appropriate means here phenyl first. So N-protected phenyl was taken in a reaction mixture. So that was treated with a free N-terminal end of the solid supported polymer. And in this reaction media, this solid support is not soluble, but free NH2 of the methionine is reacting with the reactant present in a solution. So once the reaction occurs, the peptide bond formed between the methionine and phenyl, no? So that is also, it is not soluble in the solution. So that is the reason one can filter it off there itself. So it can take out a solid. To that solid, whatever the solvent used for the reaction, that was given as a wash. So that one can remove the excess phenylalanine. So now the obtained compound is a dipeptide. So dipeptide of methionine and phenylalanine supported polymer. So that one again deprotection, removal of F mark of phenyl, and it was treated with a reaction mass. That reaction mass contain N-protected glycine. Once the bond formation between the glycine and a dipeptide, so that is also insoluble in the reaction mixture. That was filtered and enough solvent wash is given to remove the glycine. A tripeptide is obtained. So the again like this deprotection and coupling is achieved on the solid support. So once the desired peptide, and one more thing, what is here? Suppose a side chain, a hydroxy group, okay, thionine sulfur group, or indole group. Such side chains are there means those side chains is protected. O protection, N protection. Okay, protection should be made for the side chain containing functional group. So in this case, tyrosine is having a hydroxyphenol group that was protected with benzyl group. So like this we will get. So then afterwards, here itself I right. Then deprotection of solid support and then deprotection of benzyl group. Then what we will get? We will get what is called as enkephalin. So name of this pentapeptide is called enkephalin. So more particularly, it's called methionine 5. So what is 5? 5 is a C-terminal end of the amino acid. So it's a pentapeptide and the terminal amino acid is methionine, methionine 5, enkephalin. So one more enkephalin I am taking. So I will tell you the what are enkephalin. So one more terminology used. So after the synthesis of one more pentapeptide, I will tell you the difference between the methionine 5 enkephalin and one more what is called leucine 5 enkephalin. So this is the again here I am taking one more pentapeptide. So the difference between the methionine 5 enkephalin and leucine 5 enkephalin. So only the C-terminal end of the amino acids are different, but the remaining amino acids are same. And these type of uh, ones are called peptidomimics. Okay, so their peptidomimics are arises due to the biological activity. Suppose a methionine 5 enkephalin, it is not soluble in the water. If it is not showing better physiological activity, then what can do? So can change in the C-terminal end or N-terminal end to check the otherwise to mimic the existing peptide, and this process is called peptidomimics. Okay, that I will discuss after the synthesis of leucine 5 enkephalin. So enkephalins are the examples of peptidomimics. So what are peptidomimics? Peptidomimics are the small protein or peptide, so which are arises due to the small changes to the existing peptide or protein. Okay, so that is the thing you need to remember. So in the previous one and to this one is a difference is only the C-terminal amino acid. See, for example, if there is a question on, so what are peptidomimics? So give an example. So the enkephalin, leucine 5 enkephalin, methionine 5 enkephalin is the example of peptidomimics. So what are the peptidomimics? Peptidomimics are the peptides, so which arises due to the small change to the existing peptide or protein, and these are prepared or arises to mimic the existing peptide. Okay, so this is about what is called peptidomimics, and few two examples, methionine 5 enkephalin and leucine 5 enkephalin. Otherwise, sometimes they will ask direct question, how do you synthesize a pentapeptide called leucine 5 enkephalin? How do you synthesize? By employing a solid phase peptide synthesis, glycine, phenylalanine, and methionine. So you should be able to in such a way that if it is a solid phase peptide synthesis, you have to start with the C-terminal end. If it is a solution phase synthesis, you should start with the N-terminal end. So that is the thing you need to remember. So in a solution phase synthesis, protection, activation, coupling, and deprotection is the common method for the synthesis. So whereas in the solid phase peptide synthesis, C-terminal end of the amino acid. So in this case, leucine 5 enkephalin. So in this case, leucine, what is that? N-terminal end of the leucine is protected using a BOC. So what is BOC? BOC is a N-protecting amino acid. So this one is treated with chlorine. So how the chlorine? So chlorine should react with the OH of the carboxylic acid of the leucine. So there will be a removal of H and Cl. So HCl is going out. So what is the obtained? Obtained one is leucine supported, polymer supported one, resin is obtained. So this was treated with 1% HCl. So 1% HCl is the deprotection of BOC. So the obtained one is leucine, means free NH2 group. So look here, there is no BOC here. So BOC is deprotected in this process. So NH2 is free now. So this NH2 is react with the C-terminal, means react with the carboxylic group of the phenylalanine. So which was taken in a reaction mixture. In the reaction mixture, N-protected phenylalanine was taken and this solid supported leucine was added. There will be a reaction between the amino group of the leucine and the C-terminal carboxylic group of the phenylalanine. A peptide is formed. So once there is a peptide bond formation, it will float on the reaction media because it's a solid. Once there is a float, then you can filter it off with the excess solvent which was taken during the reaction. Once the dipeptide is obtained, then deprotection, N was deprotected and treated with glycine. So like that you can continue until getting a desired peptide. Once the desired peptide is obtained, you have to release the solid support. It's called release of resin. So once there is a release of resin and you have to check, so the side chain protected one. So in this tyrosine is having a side chain OH that was protected with benzyl group. So that can be removed by using a deprotecting reagent called trifluoroacetic acid. So once all the deprotection is done, now the pentapeptide is ready. The obtained pentapeptide is called leucine 5 enkephalin. Okay, so now I discussed about the synthesis, more particularly solid phase synthesis of methionine 5 enkephalin and leucine 5 enkephalin. So now I will explain about what is peptidomimics. So look here, peptidomimics is a small protein like a chain designed to mimic a peptide. They typically arise either from modification of existing peptide or by designing smaller systems that mimic the peptides, okay, such as peptides and beta peptides. Irrespective of the approach, the altered chemical structure is designed to advantageously. That means, suppose a existing peptide is showing a difficulty in the solubility. If it is showing a less active physiologically, that can be altered by changing into a N-terminal amino acid or C-terminal amino acid, or by just incorporation to the other amino acid in between. Okay, this process is called peptidomimics. Okay, So this is about, so usually they will ask, write a note on peptidomimics. Okay, so give the sentence about peptidomimics. You can say that peptidomimics is a small protein like a chain designed to mimic a peptide. Okay, and these are arises due to the uh, uh, problems of the existing peptide. So the problem may be a solubility problem. The problem may be a physiologically active one. So to make better active, to make bioefficacy, the peptide, existing peptide is subjected to modification. The modification may be a change in the C-terminal end or maybe change in the N-terminal end. Okay, so this is about peptidomimics. And we also need to discuss one more a decapeptide. Okay, so the decapeptide is called oxytocin. I think you are aware about oxytocin. So this is the oxytocin. Okay, what you need to remember? So this is 1, 2, 3, 4, 5, 6, 7, 8, 9. A nanopeptide. So in this nanopeptide, what is C-terminal end? C-terminal end is glycine. So what is N-terminal end? Cysteine is the N-terminal end. And here one more thing you need to observe. Two cysteine units are joined by disulfide linkage. Okay, this is about cysteine. So now let's look into the how this oxytocin is synthesized by employing a solid phase peptide synthesis. Solid phase peptide synthesis means one should start with the C-terminal end of the peptide. So it should start with the glycine. So that I will explain in the next slide. So look here, this is the one. So F mark means amino group is protected. Glycine part, amino group of the glycine is protected using a F mark. So that was treated with a resin. Okay, how you know that? Carboxylic means COOH. OH and H, removal of water by employing a DCC. So DCC takes the water, converted as a DCU. Then what is obtained? Ester is obtained. So ester in the sense a F mark glycine is bound to a solid support. So at this stage, deprotection of F mark. Now free amine is obtained. So this free amine is treated with the appropriate amino acid. Appropriate amino acid means it was treated with leucine. Then dipeptide is obtained, and then deprotection of the amino group, and it was treated with phenylalanine. Then deprotection, then it was treated with glycine. So like this, it is proceed to get a desired peptide. And one more thing, suppose a side chain contains sulfur means that should be protected. Maybe by using benzyl group. Suppose a side chain contain free amino group means that is also should be protected by using N protecting group. So once there is a desired peptide is obtained, then release of solid support. After the release of solid support, then what is left over? Left over is N protected one and side chain functional group protected one. So side chain protected one and N protected one is released. When N protected one, if it is a F mark, it is released by using 20% piperidine. And disulfide linkage is made by using potassium ferricyanide. So this is an oxidizing agent. It converts SH to SH to S single bond, S bond, disulfide linkage. So disulfide linkage is formed by the reagent called oxidizing agent. It's called potassium ferricyanide. And what is obtained? The obtained one is oxytocin. So there are questions on this oxytocin. Write the structure of oxytocin. And also there are questions on oxytocin. So give the solid phase peptide synthesis of oxytocin. So then you should aware about this structure and you should able to write this process. C terminal end you have to take, make the solid support with the DCCHOBT, and then it was deprotection and then treated with appropriate amino acid. If at all a amino acid contains side chain functional group, those should be protected. Okay. And at the end release of solid support and then release of side chain protected and N protected, and it was treated with the oxidizing agent to make a disulfide linkage. So this is about the solid phase peptide synthesis of oxytocin. Okay. uh, with this, your amino acid and peptide syllabus is complete. So that means unit 4. So what is unit 4? Unit 4 contains vitamins as well as amino acids and peptide. So that unit 4 is completed. Of course, there are question and answers. Maybe let's look into that. So let's go to the question and answer discussion. So look here. How do you, I think all the question and answers I have given in your notes, no? So what you can do, maybe you solve all these question and answers, okay? You write in a separate notebook, okay? So that notebook, in that notebook, whenever you are coming to the offline classes, no, so that time you show it to me. I will correct it if it is anything, if at all, if you have written anything wrongly. And since the one unit is completed, so at the same time, in your free time, just have a look into this. So maybe we will also go for a internal for unit 4. Okay. And let's look into the questions. So how do you achieve the activation of carbonyl group using DCC? That means you need to write the structure of, so DCC is the reagent. It is used for the activation of carboxylic group, and also it is useful for the coupling reaction. So how means you have to write the carboxylic group. That carboxylic group is OH is there, no? The lone pair or the bond pair of the carboxylic group of the amino acid react with the DCC. How it is reacting? More particularly carbo, means the carbon which is attached to between the two nitrogen. Okay, to that it will form a O-acyl urea. So what we can say that amino acid, carboxylic end of the amino acid react with the DCC and convert it as a O-acyl urea. So in this way, the amino acid is activated. So maybe you need to write the word called O-acyl urea. Okay, so that you need to write. And write the full structure of tripeptide. So tripeptide containing alanine, leucine, lysine. So that means you should aware about what is alanine, what is leucine, what is lysine. So in this case, alanine is the N-terminal end, lysine is the C-terminal end. So you have to write the structure. So leucine is the in-between one. So if you are changing this order, you may not get the marks. So left side alanine, and center leucine, and then last is lysine. So you should know the side chain present in the alanine. CH2 phenyl is the side chain present in alanine. So what is leucine? Isopropyl group. What is lysine? Uh, CH2 three times NH2, that side chain. Okay, so that you have to write. Amide bond between the alanine and okay, carboxylic end of the alanine and amino group of the leucine. Carboxylic end of the leucine and amino group of the lysine. So here C-terminal end is free, here N-terminal end is free. Okay, that's what you need to write when you are writing a structure. Then what is CNBR? CNBR is a reagent called cyanogen bromide. How it is useful in the cleavage of peptide? So that means you know that one particular CNBR particularly cleaves the peptide containing that amino acid you have to mention. What is that amino acid? Is it a tyrosine? Is it a tryptophan? Is it a methionine? So that you have to find. How you can find? Maybe just to look into the notes. Okay, then write the synthesis of oxytocin. Just now we discussed. Write the synthesis of oxytocin, emphasizing protection, deprotection, and coupling. Protection, deprotection, and coupling. So this is based on the solid phase peptide synthesis only. Okay, so because here deprotection and coupling words are there, no? So based on that I am saying deprotection and coupling is achieved on the solid support. So protection means N protection. Okay, and give the reactions and its mechanism in the cleavage of peptide bond using CNBR. Again it's asked. Okay, CNBR particularly cleaves the what is called as methionine containing and the adjacent amino acid that you have to mention. So 2 marks, no? Just mention the in terms of words you can mention. If you are able to write the structure, that's also a fine, but not the mechanism, because it is asked for 2 marks. If at all, if it is asked for a 3 marks and 4, you can write the complete mechanism. Then outline the synthesis of alanine. So alanine synthesis, how alanine can be obtained from malonic ester, or maybe a Gabriel thalamide method of alanine synthesis. Okay, those things you can mention it. So with this, I can go to the next slide. So what are the peptidomimics? So just now I mentioned that peptidomimics are the small molecules or the peptide arises due to the changes or mimic the existing peptides. For example, enkephalin. Enkephalin means methionine 5-enkephalin or leucine enkephalin. Islate means, so just by altering N-terminal end or C-terminal end, so if it is increasing the bioefficacy or if it is increasing the solubility, so that will be a good thing. That will be the utility. So Sanger's and Edmonds method. Sanger's method means dinitrochlorobenzene is the Sanger's reagent, and Edmond reagent. What is Edmond reagent? It's thiophenyl cyanate. No, phenyl thiocyanate is the Edmond reagent. Okay, both the method you have to mention. So to find the N-terminal end of the amino acid is useful for the Sanger's method. By employing Sanger's method, so one can find out the N-terminal end of the amino acid, but not the sequence. Whereas the Edmond method, it will give you the sequence. Sequence in the sense how the amino acids are arranged. Okay, whether they are arranged in phenyl glycine alanine or alanine glycine phenyl. So that sequence can be find out by employing Edman method. What is Edman method? In this method, phenyl isothiocyanate is treated with the peptide or protein to find the sequence of amino acid. So what it will do, it selectively convert N-terminal end of the amino acid as a identine. Okay, when it is hydrolyzed, N-terminal end of the amino acid is obtained as a identine. The identine was characterized to find the name, what is the amino acid. Okay, so similarly, the shortened peptide again treated with phenyl thioisocyanate. Then shortened peptide N-terminal end is converted as a identine. In this way, one can find out the sequence present in a given peptide by employing Edmonds method that you have to mention. Then illustrate the use of DCC-HOBT in the peptide synthesis. This is called HOBT method. Okay, what is happening? The carboxylic end of the amino acid react with the DCC, forming a O-acyl urea. To that O-acyl urea, HOBT is reacting, forming another intermediate. That intermediate is attacked by the N-terminal, means free amino group of the amino acid, and thereby leads to the formation of peptide. So that you have to explain it with the example. Right. Then write a short note on solid phase peptide synthesis. So in this solid phase peptide synthesis, what is called deprotection and coupling is achieved on the solid support. In this method, one of the reactant is taken in solution as excess. And in this, once the bond formation between the active amino acid part of the solid support and the amino acid present in a solution, so that was filtered and washed with plenty of solvent to get rid of the impurities. And then it was subjected to deprotection and coupled with a appropriate amino acid to get a peptide. Thereby one can reduce the time consumption and more steps involvement. CNBR again it is asked. Maybe you just look, see peptidomimics. Then how the following deprotected, explain the mechanism. So this is what is called as a dipeptide is given, means what is called as a amino acid is given. This is one amino acid. So in this amino acid, C is also protected, and what is called N is also protected. And what is N protected? Its N protected one is a bark. So bark can be deprotected by employing a trifluoroacetic acid. So that you have to mention trifluoroacetic acid. It will cleave here. It will also cleave here. It will also cleave here. That you have to mention. Means carboxylic acid is the one of the byproduct. Isobutene is another byproduct. And also you have to mention the C-terminal deprotection, means carboxylic acid deprotection and N deprotection. You have to mention. Since it is a bar, trifluoroacetic acid you can use. And you don't know about the R dash. So you can, if R dash may be ethyl or methyl or benzyl, then you can subject it to a reduction, hydrogenation. So then this will be break. Then give the synthesis of phenyl. So this PHE stands for phenylalanine by azolactone method. So how the azolactone is obtained, and how using azolactone, how phenylalanine is synthesized, that you have to mention. So by taking a suitable example, explain the peptide bond formation using DCC. So DCC is the method. By employing this method, one can do a activation and coupling in a single step. Okay, so you have to write the N-protected amino acid that was treated with DCC, form a O-acyl urea. That O-acyl urea treated with unprotected amino acid. So like this, you can write the answer to this all this question, and uh, 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 whenever you are coming to the college, that time you show me that, and at the same time you prepare for the internal. Okay, so that internal is for since it is a 13 hours, so 13 plus 13 and half, maybe you can prepare for a 30 marks question. Okay, so this is about of course few more questions are there. This I have discussed. Here, look here. So what is this? Outline the solution phase synthesis of leucine, valine, glycine. So here a solution phase is mentioned. So solution phase mentioned means one should synthesize starting with the N-terminal end. Here the N-terminal end is leucine. So leucine is what you can say that you take N-protected leucine that was subjected to activation. Activation means treated with thionyl chloride. So using thionyl chloride, carboxylic acid is converted into Cl. So that was treated with the C-protected valine. So there will be a reaction between the amino group of the valine and acid chloride group of the leucine. A dipeptide is obtained, and then deprotection of the C, and then activation of the C, and then it was treated with the C protected glycine, and then D protection of C protection as well as N protection. So that is how a tripeptide is obtained using a solution phase. Solution phase means you have to start with the left hand side amino acid. Solid peptide synthesis means you have to start with the right hand side amino acid with the resin. So that difference you need to remember. So next, again the Edman method is there. So you can get one question from the Edman or Sanger's method. Edman or Sanger means you will get one question on it. So synthesis of amino acid, you will get one question there. So methods used in the peptide bond formation, there you will get one question. Solid phase peptide synthesis, there one question you can expect. So these are the key parameters. And then protection group, C protection and N protection. So N protection group, you will get one question. Okay, so these are the things you need to remember in this amino acids and peptides, and give the steps involved in the conversion of malonic ester to alanine. So this is what something like how the alanine is obtained from malonic acid. So steps involved you have to write. First is about what is called as alkylation. So alkylation means treated with base and treated with methyl halide. Then what is the thing you need to do? So you selectively you have to hydrolyze. Okay, and then treated with what is called as azide. Then courteous rearrangement, and then you will get a carbamate hydrolysis. So you will end up with the alanine. Then here write the structure of F mark. F mark means aspartame aspartame amino group is protected using the F mark. In the aspartame, side chain is protected using a benzyl group. Carboxylic acid is the carboxylic part is the carboxyl part of the asparagine, and so that means you have to write this structure of the aspartic. Here, amino group is protected with F mark, and the side chain is protected with benzyl, and the carboxylic acid is free only. So that structure you need to write. Here the serine. So serine means OH is the functional group is present. That OH is protected with the tertiary butyl group. And amino group is protected with Z. Z, what is Z? Z is a benzyl oxycarbonyl group. And carboxylic acid is free. So these two structure you have to write. So here the examiner or valuer is checking your ability to write the structures of N-protected amino acids or the side chain protected amino acids. So CNBR again it's asked. Explain why cyanogen bromide. CNBR is nothing but a cyanogen bromide. Okay, explain the synthesis of oxytocin that we discussed. Sketch the Merrifield synthesis. So here a tripeptide is given. So Merrifield means it's a solid phase peptide synthesis. So what you need to do, you have to take the cysteine. So in the cysteine, a sulfur is there. Sulfur should be protected with benzyl group. So, and afterwards, the cysteine, N protection to the cysteine and S protection to the cysteine, and that was treated with the solid support. Once there is a solid is support is bound to the cysteine, then deprotection, and it was treated with glycine. Then deprotection, it was treated with alanine. Then release of solid support and release of side chain, sulfur protected and alanine N protected. Then you will get a solid peptide synthesis mediated a tripeptide synthetic method. And then, so racemization. Explain how the racemization. So I think racemization arises when we are using a strong base. If there is a obstruction of proton, so then there is a racemization occurs. Okay, so if we are using a mild base, no need of worry about the racemization. And also you need to discuss what are the methods involved. Synthesis of a peptide by using azide method. Okay, azide means N3, means Curtius rearrangement. Okay, then explain the Hedman. Okay, Hedman means to find the sequence of amino acid. So this is about what is called amino acids and peptides. So maybe you can just solve all these questions in the sense you write in a separate notebook, and when you are coming to the college you show me that. That is one thing. And one more thing, you at the same time you prepare for a internal test for this vitamins and amino acids. And maybe in the next class I will start one more unit. Okay. And maybe if you have any doubts, you can ask me. If there are no doubts, you can log out.